Welcome to my presentation. Today we'll have a discussion on the topic methods of studying animal behavior. Before looking into the methods of studying animal behavior, first we'll see what is behavior is. So behavior is everything an organism does in response to external stimulus as well as internal body states. Generally, it is of two types: innate, unlearned, instructive or native behavior, and learned or acquired behavior. The scientific study of animal behavior is termed ethology. The word ethology is derived from two Greek words, namely ethos, which means habit, and logos, which means study. This picture, it deals with a variety of behaviors seen in insects like ants and dolphins and caterpillar, animals like chimpanzees, kitten, and the birds which build the nest. So why we have to study animal behavior? For example, the basic knowledge on feeding behavior will help us to know what is the type and amount of feed that animal need, feed conversion efficiency and control of feeding. And next one, the basic understanding of the behavior of livestock will facilitate handling, reduce stress, and improve both handler safety and animal welfare. So, what type of behaviors are recorded while studying animal behavior are present given in the slide, which includes feeding, drinking, blinking, walking, fighting, flying, territoriality, dominance, hierarchy, mating, nest building, parental care, social organization, daily or seasonal activities, orientation, migration, mother-infant relationship, inter and intraspecific relationship. So while studying the animal behavior, these kind of data are recorded, which are state and events. So state is the behaviors that occur for an extended duration, for example lying, walking, foraging and sleeping. And events, these are the behaviors that are short in duration and generally counted rather than timed, for example fighting, yawning, sneezing, vocalizing, etc. So now let us look deep into the topic. What are the methods of studying animal behavior? The mostly accepted wild methodology was proposed by Altman in 1974. There are actually two methods of studying animal behavior, which includes the study in its own natural environment, which is in wild or in natural habitat method, or the study in which is performed outside of its natural environment, which is the study in laboratory or experimental method. This method is generally dealt with in relation to brain and its physiology. These 6 steps are various methods of studying animal behavior in wild or in natural habitat method. Which is the ad libitum sampling, focal animal sampling, scan sampling, all occurrence sampling, sequence sampling, 1-0 sampling. So ad libitum means at one's pleasure. It is the simplest way of recording animal behavioral acts. Here no systematic constraints are placed on what is recorded and when. The observer records whatever they can see and that they think is relevant at given time. Because of this, it is an informal, non-systematic and often used in field notes. Since the observer cannot keep track of everything that is going on, the results of these observations will always be biased by the behaviors, individuals or situations that most attract the observer's attention. Next is focal animal sampling. This technique is useful if the observer is interested in what a particular individual is doing over a given time and can used to be record states or events. This is the most satisfactory approach to studying groups because the observer focuses his attention on a single individual so he can provide accurate data on frequencies and durations of studied behaviors. Next is scan sampling method. It is the best method suited for recording behavioral states. Here, a whole group of subjects is rapidly scanned at regular intervals and the behavior of each individual is recorded. A single scan may take anything from a few seconds to several minutes depending on the size of the group and the amount of information recorded for each individual. Next is all occurrence sampling. In this sampling, the observer watches the whole group of subjects and records each occurrence of a particular behavior, describing the context in which it occurs as much detail as is required. This technique is mainly used for recording rare but significant types of behavior where it is important to record each occurrence. Sequence sampling. So this sampling period is from start to end. The focus is on a chain of behaviors, for example, courtship display in sticklebacks. Next is 1-0 sampling. Here, if behavior it is noted, it is if a particular behavior which is recorded is noted, it is given as 1, or if does not take place, it is given as 0. The observer scores whether behavior occurs 1 or not 0 during a short interval of time. This method it is suitable for recording states and events. Next is the study in laboratory or experimental method. This includes 3 methods which are neuroanatomical, neurophysiological and neurochemical. Neuroanatomical. So neuroanatomy, it is the study which is related to the behavioral study in response to the physical structure of brain and its parts. Mostly the brain parts are destroyed and their function is deducted from the deviated behavior. This is achieved by making cuts with a knife, injecting neurotoxic chemicals like cyanic acid, placing small and precise injuries in the brain using stereotaxic equipments. This is the oldest and crude method in neurological research in studying the relationship between neural structure and behavior. This generally includes 3 main headings, which are classical experiments on rats and discovery of Broca's area and the discovery of Wernicke's area. So first is the classical experiments on rats. This was performed by Carl Lashley on what he did is he conducted a series of influential experiments in the early 20th century to investigate the neural basis of memory. Methodology. What he done is the rats were trained to navigate complex mazes to reach a food reward. Following successful maze laning, specific areas of cerebral cortex were surgically removed. Next, the rats were then reintroduced to the maze to evaluate the impact of brain lesion on their previously acquired memory. So the key findings of his experiment are, he observed that extent of memory impairment correlated more with the size of cortical lesion than its specific location. This led to the principle of mass action, suggesting that memories are distributed across the cortex rather than localized to a single area. Next is equipotentiality. Lashley also found that if one area of the cortex was damaged, other cortical regions could sometimes assume its final role. This phenomenon is termed as equipotentiality. So next is discovery of Broca's area. So first, the Broca's area was identified by Paul Broca. He is a renowned 19th century French physician and an anatomist. So what is the Broca's area means? It is the region in the left frontal lobe crucial for speech production and motor speech area. This is also called motor speech area. Clinical observation. He was a doctor. He observed a patient with a head injury would ultimately suffer with a defective speech. So what is the conclusion he made is damage to the Broca's area of the brain can result in a significant impairment in the ability to produce speech, a condition known as Broca's aphasia, which is characterized by difficulty in forming words and speaking fluently while still largely retaining the ability to understand language. Broca's area is responsible for motor planning of speech. Next is discovery of Wernicke's area. This area was identified by a German neurologist called Wernicke in 1874. So what his clinical observation was is Wernicke, he noticed that brain injuries in the left posterior section of the superior temporal gyrus caused aphasia. Aphasia is a condition that impairs language comprehension and prediction. Wernicke hypothesized that this area was responsible for reflexive mimicking of words and syllables. The conclusion is language processing is a complex function involving a network of brain regions, not just Wernicke's area alone. Next, here I have mentioned what are the limitations of doing the neuroanatomical techniques. Next is neurophysiological techniques. So these techniques are used to study animal behavior by recording the brain activity or stimulating different regions of brain. There are two key neurophysiological methods, which is recording of brain activity. First one is recording of brain activity, and second one it is brain stimulation by stimulating different areas of brain. The recording of brain activity, it is performed intracellularly and extracellularly. So the first successful recording of human brain activity was performed by Hans Berger. He was a German psychiatrist. He done this with the help of a specialized instrument. What he invented is electroencephalography, which is a method used for recording the electrical activity of brain, commonly described in terms of brain waves. So these 5 type of human brain waves are recorded in an electroencephalogram, which are gamma, beta, alpha, theta and delta waves. Next is brain stimulation experiments. Brain stimulation experiments in animals involve stimulating specific brain region with electrical or chemical agent to observe the resulting changes in brain. For example, it is given electrically, chemically and with the help of light. This brain stimulation experiments are often used to study a wide range of behaviors, which include motor behavior, sensory perception, learning and memory, emotion and motivation, and social behavior. Next is neurochemical technique. This technique is also called chemical brain stimulation technique. Here, the study of chemicals that influence the function of neurons with respect to analysis of behavior is done with respect to analysis of behavior. In this method, the neurons are affected more selectively in their behavioral activity by the local application of drugs, neurotransmitters, blocking agents, and hormones. More recently, the brain is being explored for its various functions using positron image tomography. So, the two important classical experiments are performed by Fisher and R.P. Mitchell, which demonstrated that sexual and maternal behavior can be induced by injecting minute quantities of sex hormones into hypothalamus, and the other is administering the estrogen in anterior hypothalamus of brain of female cats when not in heat produce the mate. On this contrary, if the same hormone was injected in some parts of brain, similar behavior was produced. So, it is not that much easy to perform or to study the animal behavior through these methods. So, there are many challenges in studying animal behavior. The key challenges include observational difficulties, interpretation and bias, data collection and analysis, ethical consideration, and species specific challenges. For example, animals may exhibit important behavior in concealed locations, making observation difficult. For example, nocturnal animals. Species specific challenges include nocturnal animals and aquatic animals. Studying these animals that are primarily active at night can present logistical changes. Observing underwater behavior often requires specialized equipment and techniques. So this is an overview of how the animal behavior is studied in animals through various methods. Hope this video is useful for every one of you. Thank you.